That's more manageable. Hello, everyone, and welcome to the first late, if only by 3 minutes, Roach Crossing weekly live stream. Thank you for your patience. I usually shower and clean up a little bit before the live stream, get some of the daily dirt and gunk and grime off. And we've had a surge in the humidity because it has been raining on and off, little showers all day. And so got out of the shower and thought, oh yeah, I have all kinds of time to dry off, and struggling to get socks on because of how moist and clingy everything is. So, but I am here, and again, I have an order from a plant place has shipped. So, hello, seeing a couple of folks who haven't been around in a bit. Tennyson, I think you were here last time. Alex, good to see you. Hylas, good to see you as well. I'm not really sure. I'll always see the last, maybe Shayna. Nice to Chain. Nice to see you too. Again, the last couple of live streams, I've been kind of hard pressed to have projects on my mind or things directly to talk about because of I've been mostly the last maybe 5 weeks have just been slammed with stuff I have to get out, and this week was particularly intense. I spent Monday, Tuesday, and Wednesday sending boxes. And one of those boxes was a complete flunk failure on my part. Maybe I'll talk about that. Let's see, test. So I have plenty of questions. Don't worry. Feel free to ask. You have my fullest attention right now. I don't know how long this particular stream will go. I don't have any pressing things to attend to today. Finished up husbandry of all the roaches and isopods too. That kind of got stretched out over the course of a week. And so we wrapped that up today. Have some bad news. I've lost my Blabberus colossius Peru colony. That's been with me since maybe 2011, 2012. I put them in a upgraded container. I put them on more heat because I wanted to get them available in good numbers, because I have Ecuador and Panama doing very well nowadays. In fact, I think for the last maybe year and a half, 2 years, anytime anybody's messaged me asking for Peru, I've told them, hey, I don't have Peru right now, but I had Panama, which is basically they are identical in size and coloration, all this other stuff. But now I have to track down somebody who can guarantee they have some Colossius Peru that's pure to send back to me for me to restart the colony. So that's probably not going to be fun. But if anybody here has it or knows of anybody who has Colossius Peru and it can guarantee that it's pure Peru, let me know. But what was I going to say? So that was one L from this week's husbandry stuff. Quite a few things are doing well. I was kind of maybe almost on the verge of losing my Paranophyta formosana at the end of last fall, and then I took extreme corrective measures, and that colony's back up to getting ready to thrive again, so that's good. Anything else? Any bins I dissolved? Not really. A lot of the millipedes I've gotten wild caught and whatnot the last couple of months are still alive and doing well. It's just a matter of waiting for babies, which I will just keep doing. There was a loss in some uriards I received from a friend in Tennessee who I think I got like 7 of them total or so. I put them in the enclosure, and I used wood mixture that was too old, or I didn't do it ideally, and I kind of half-assed it, and instead of fermenting and becoming infested with mycelium, which is really good for millipedes, the substrate ended up becoming putrid. It might have even been anaerobic decomposition, and it killed all of them. But fortunately, he will probably stumble across more eventually. and the other cultures of those are doing well. Again, no babies yet, but there probably will be. Let me take a moment and read through what we got in the chat here. Tarantula Jake says, I missed the last one, and the one where you were wearing a bug net I was watching while driving home and the tire fell off the truck. Oh boy, on the interstate. That doesn't sound like any fun at all. Edison says, it reminds me, I recently asked my earth science teacher about some genetic stuff with isopods I noticed on Instagram that around the same time a lot of people was panicking and closure started throwing orange individuals. I think it's probably a switch gene activating from either light or altitude differences. He also said he thinks other Q-bars may throw switch gene morphs. If that is the case, what do you think about this? He studied genetics for 2 years in college, but some of it was out of his scope, so if you're not sure. So I don't know enough about genetics to say if that is particularly the case. That might explain a lot of things if it is. But a lot of people, what I think happens is these imports of stuff or groups that get distributed all come from a handful of people or places originally, and so you've got a lot of individuals that are very closely related to each other. And sometimes, maybe it's a handful of individuals that were brought into Europe or something, and then they mated, for a couple of generations, and then they're distributed. But basically, the reason why it seems a lot of these all pop up at the same time is because at some point in this process, that allele mutation for, it's probably just simple recessive orange in the panda kings, the red pandas, or whatever they're calling them. It probably just it popped up in because of the smaller population at first, and because everybody's are related and everybody would have gotten about the same size, a cohort of individuals from a similar source around the same time, then they get distributed, and then all of a sudden everybody's next generation starts throwing that color. Well, it's because you had those alleles in that population that were just sort of sticking around, and now you've got a 5 count or an 8 count or whatever, and the siblings doing each other, and then you get simple recessive orange. The allele can finally surface in the colony. So I see it's not necessarily that something similar is happening in all those places that's causing them all to turn out that way. It's more of a question of, where did the stock come from and what's the history of it, because that would explain why everybody starts getting the same mutation at the same time. Chain says, ever heard about juvenile amblypygids being able to have a brood? Not sure if they were just juvies, but P. whitei with white dots only a few centimeters long. 2 out of 6 birth within the first couple weeks. Just find it weird, but maybe I just haven't seen enough yet. So going off of kind of conversations I've had with Alan, and Alan keeps a lot of amblypygids, is that they can molt multiple times after they've already reached a reproductive size, kind of like how isopods can do that too, or a lot of isopods can. And so you may have just gotten adults that were small because of environmental conditions, and then they reproduced and they will molt again. But I think Alan said that amblypygids don't have a mechanism to store sperm long term, so they will need to be, at least if what I'm remembering from my conversations with Alan are correct, they will need to be mated again to make, if you want to get more fertile eggs and more offspring out of them. So yes, Alan, not the picopods that I have had for a while, but ones that he ended up giving me in a culture, a mixed culture of a bunch of other stuff, have been identified. I can't remember what the Latin was. It hasn't sunk into the memory banks yet, but if I ask Alan, he'll be able to pull it up. So, but yes, they have been, at least one type of them has been identified. I'm still trying to salvage the original species or strain, and it's a very convoluted method that I'm using to try and find some of them in the vastness of the old enclosures, refuse and whatnot. And so I'm currently in the middle of doing that, and I don't know if it's going to go too well, but again, since I have some of that other culture he gave me, we will not lose at least one organism that properly bears that common name. Tennyson says, I didn't think about that. Maybe it will make an interesting project someday. Oh, with the alleles just sort of popping up at the same time. I try to keep some closely related P-scape or lighting mixes in different light setups and see if the genetics stay similar to this. No, I can tell you already that all of the simple recessives that are in the lottery mix, you can't switch them off. Like it's this the odds of that reversion to the wild type in at least in the simple recessive oranges for sure is ridiculous. It's I think it's might even be for various reasons higher than the chances of things mutating in the first place into the simple recessive orange. So anybody who says that they have a colony of oranges or Dalmatians or whiteouts or any of those simple recessive mutations, and then suddenly starts finding, oh, I had like a bunch of them. Oh, they always throw this. Oh, they always throw these wild. Oh, they always throw this. They either have compromised stock from the beginning, or they have isopods getting into their bins from other containers. So, but recessives can vary. I've seen it happen once, maybe back around 2011 or 2012 in my Spanish oranges. I had the bin completely separated from all my other cultures, which were in locking or not locking, snap-on lid hexagons that I used to keep things like German roaches and whatnot from getting out. So those were all my cultures of wild types and whatnot were like that. I used at the time because I was not aware of the whole rotting leaf litter and all that other stuff, and I was terrified of introducing pathogens at the time. I was using bone dry oak leaves to feed them, along with the other typical isopod diet stuff that I had stored in my parents' basement. And so I remember opening up the bin and going through the colony, and at some point finding in one going about 3 wild type individuals that popped up. They were all males, and I did pull them and I tried to isolate them, but they might have been sterile actually. But this was in pure Spanish orange stock. There is absolutely no way outdoor isopods could possibly have gotten into that culture given my protocols at the time. And so it can happen, but that's the only time that I've ever seen simple recessives revert to wild type coloration, or rather to dominant coloration. There's 3 males that may have been sterile in Spanish orange, and I've never, ever, ever seen that happen in any other culture ever since then. So that alone probably puts the odds at somewhere in like 3 in like several million. And I know there are a lot of people who reproduce isopods, but again, that's the odds of 3 in several million, or one in a million, or even more than that, is not like, oh, if you have a million isopods, it's going to happen to one of them. It's every single time that there is a reproductive event, it means that there is a 1 in a million chance at that event that something like that could happen. So, but anyways, Tennyson asks, would you want to do that with pure wild type stock, or would it not work at all? Need to see if some of the mutations or switch genes being activated. So they've done this sort of stuff for a very long time with plants. It's a little bit of a stretch, but there are different chemicals they'll subject plant embryos to, or plant seeds to, that will cause them to mutate in predictable ways. I can't remember what the chemical is called. It's a carcinogen in humans, I think. Well, I guess it is in plants too. But the most famous example, or one of the most famous examples, is taking Rudbeckia hirta, the common black-eyed Susan. They expose the seeds to this certain chemical. I can't remember. I'm gonna look it up. Ploidy, and see if I can find it. Colchicine. Colchicine seems to be the chemical. Oh, I bookmarked the page instead of searching it. Darn it. Colchicine? I don't know how to pronounce that. Oh, is a medication used to treat gout, Behçet's disease, and and a bunch of other miscellaneous things. Historical treatment for joint swelling. It's a chemical found in the autumn crocus. Okay, I guess I was completely off base with the carcinogenic thing. But anyways, oh, certain things can happen, organ failure and dysfunction after consuming too much of it, blah blah blah. Anyways, so they would take the seeds and subject them to this, and it would cause their chromosome numbers to double. So they go from diploid to tetraploid, and so that, it does alter the phenotype of the organism, becoming tetraploid. They gain red markings on the petals, and the overall size is bigger than the wild type stock. So, but anyways, but the point there is that environmental things can be mutagenic to different organisms, but you have to find something that doesn't kill the isopods, and you'd have to administer it in a way that would affect the developing offspring, or affect the developing embryos to do that. And then there's also not. I see what you're saying there, Tennyson. Also with the light difference and whatnot, there are also epigenetic things that can happen, and some of those are partially heritable, and that's a very novel, ongoing sort of field, is looking into the heritability of gained traits, which is funny because hundreds of years ago, before evolutionary theory, that was the primary. Supposedly, this could be also, oh, we're so progressive now, we know so much more, but people have known forever. Oh, your kids, if they're actually your kids, are going to look like you because you made them, basically. But that, which is basically how heritability and stuff works in the simplest way. But anyways, but there was the supposedly the antiquated thing with, like, oh yeah, if you like you cut off like a mouse's tail and you breed the two mice together, they'll have shorter tails. The idea that acquired traits could be passed on in that sort of manner. And we now know that that is not true in that overly simplified sense, but now we're also finding out that certain things can be switched on, and that your offspring can inherit those things, or certain stressors or chemical exposures or whatever can actually be directly inherited by your offspring genetically. So it's kind of interesting how that's come full circle with some nuances. But I have noticed that dark and light isopods, um, polar isopods tend to be darker in color, and they tend to retain that for a while, even when warmed up. So there's something going on there. I mean, it could be just a physiological change, and that drier isopods, at least in like Porcellio scaber, tend to have better pigmented markings. A lot of this has just been from anecdotal stuff that I've done with Porcellio scaber Dalmatian, because people used to complain to me at shows that they bought Dalmatians, not necessarily from me, but they would buy Dalmatians and then they would all revert to white or something like that. And I remember these one or two people who came out, they were like brothers or something like that, maybe. But they were telling me about how they had like tons of them. They were doing well, and they lost all their black spots, and then the colony just crashed and died, and they were all gone. And that made me think, when I was trying to, because after the premise for this is that I've tried for many, many, many years to breed for larger, consistent spots in Porcellio scaber on the Dalmatian, thinking that the spots themselves were heritable. And so I did many, 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 many inbreeding projects with Porcellio scaber pure Dalmatian stock to try and breed for larger spots. And there was never any consistency to the results. Like, I could cross the father to his sibling that was also his great granddaughter, and some individuals would come out with a mixture of spots, and some would come out with, there was no consistency in spot size. It's not like, oh, I keep breeding them and the spots are more numerous and they're bigger, etc. There didn't seem to be some sort of modifier at work there with this. But then, having thought back to that story where they lost all their spots and they did really well and they died, it sounded like they were keeping them very hot and very humid without much ventilation. and that was probably why they died, is they very quickly exhausted the resources within the bin. Whatever was originally put into the setup, like coconut fiber and leaf litter and whatnot. And then because they basically ran out of food and any microbiota that was going in there, they just all died because it was so hot and humid that they starved. So that's the thing with the heritability and stuff like that. And I do think that I have had hope that crossing Dalmatian into other lines of Scaber, more distantly related lines of Scaber, that due to actually this ties in very nicely with the blog post that I put up a couple of days ago, which I can talk about. This ties into some evidence that different genes or different alleles can express differently in different lines of Porcellio scaber, and so the best example of, or the best sort of example for this, and this is, we have to keep in mind that multiple phenotypes, you can have a very similar phenotype between a bunch of different things, but the genotype can be very different. Oh, hi, Kai. Kai's here to be a thorn in my side and be a contrarian. Isn't he cute? Anyways, so imagine that you have what's a good organism to use? Imagine you have a mouse. You have one mouse that's just brown mouse colored, and then you have another mouse that's like a piebald. So there's white markings amidst the mingled with the brown markings. And so if you were to breed albinism into either of these lines of mice, the one that's solid colored or the one that has the piebalding color, and you were to look at the phenotype, because of the way that the albinism affects the pigment distribution, and because of the way that piebald is already affecting that, you will end up with mice that are all white with red eyes, red-eyed whites. In terms of the phenotype, you wouldn't be able to tell without crossing those to test stock to examine, what their genotypes are. You would never know that the albino mice that you have, that some of them are piebald and some of them are just a solid colored wild type agouti. So this carrying over to isopods is that sometimes, as seen in the cross that I have noted in the blog on 525 and May 25th, what this means is that what I think can happen is, with different strains, you can get different expression because there are other genes at play there and other alleles that are not present in the original stock that you're using, that can then influence the distribution or appearance of the other genes in your pre-existing stock. So basically what I'm getting at here with respect to Dalmatians is, I have hope that by using different lines from different places, I might be able to get a strain that ends up with a heritable component to the Dalmatian markings. Thank you, Hylas. Yes, polygenic. Anyways, so that was just kind of a very simplified explanation. But the thing here, and what I talked about in the blog, is that there is now very strong evidence that this can be the case with simple recessive mutations. So I had, everybody can go and read the blog. I'll put in the comments here for people to go and look over who are rewatching over the stream and whatnot. But basically, I had what appeared to be a whiteout pop up in a strain that I had going of scaber that has both lava and leucistic. It has lava, which is actually locality, so it has pure Spanish scaber genes. And then it also has, having crossed into that, some California Porcellio scaber stock. So who knows if they originally came from Spain? Who knows if they originally came from the United Kingdom or where? But the point is that they've been isolated. The populations have been isolated for some period of time, not just since they jumped continents, but perhaps on a geological time level. So, anyways, so I had this individual that looked like a whiteout pop up in this cross between these two strains, and my first thought was, well, like I thought that oh, cool, a whiteout, like, oh, that those alleles were hiding here. But then I thought about it some more and realized, oh, this is the stock that has the leucistic allele that is floating around in the background in it. So that's probably what this is. But the thing about the leucistic, whatever this leucistic allele is that the isopods end up with black eyes and white bodies, versus in whiteout, the whole body turns white. There's no eye pigmentation. So we had what was scoring as basically a whiteout in a strain that had no history of whiteouts. And whiteouts can pop up in anything. They've been popping up in tons of Nasodillo and Cubaris lately, so it wasn't completely out of the question. But I wanted to know if this individual that was phenotypically a whiteout would, if crossed with a leucistic, to get I wanted to confirm whether or not that was the whiteout mutation or if it was the leucistic mutation going on in this stock. And so I isolated the female and I raised her solo, and then I added a male from the leucistic and crossed the two of them, and found out once the babies got to be of good size that it's the leucistic allele, because all the babies had black eyes. So, and if these were on the same locus, if these two alleles were sharing the same locus, I would expect more of a mixture of phenotypes here in the F1. And if they don't share a locus, then we would have basically two separate mutations, and so all of these F1 offspring would all be heterozygous for whiteout and for leucistic if they were two separate traits. So basically, the nail in the coffin for this cross was finding that all the offspring had the dad's phenotype, the traditional California stock Lucy line leucistic, and so that confirmed that this was the same allele responsible in both of these lines. But again, the lava cross had completely white eyes. So if you were to just see that pop up in a colony of lavas, you think, oh, this is whiteout and not leucistic. So what this means is, and this sets a precedent, is that these simple recessive mutations, which people were saying, oh, it doesn't matter what lines they're in, oh, it doesn't matter, like, oh, you should always just bring it for the mutation, is that different strains, and in this case, different localities even, because the lavas pure lavas crossed with pure Californian scaber was the cross that originally created the whiteout looking leucistic. It means that different localities can have different expression of simple recessive genes, and that the phenotype is not reliable without doing test crosses for things. So that means that, and this makes sense because you can see this across different species of isopods too. We have the simple recessive orange mutation in Armadillidium vulgare, and Armadillidium nizatum, in Porcellus scaber, and a bunch of stuff. And the phenotype is very variable. You get the coloration in Armadillidium nizatum can be more reddish. It's more concentrated around the areas where there are pre-existing concentrations of pigment. In vulgare, it becomes basically the whole body becomes solid orange or reddish, except for the yellow markings that the females have and whatnot. And in Porcellus scaber, in U.S. orange, because the basal stock is primarily solid-bodied individuals instead of calicos or any other mixture of colors that you find in Spanish stock, you get pretty much solid oranges. So it makes sense, but this is very drastic because the main difference between the leucistic allele and the whiteout allele is the eye coloration. It also has some subtle effects elsewhere, like leucistics do not yellow with age like whiteouts do. You can find more yellowed whiteout Porcellio scaber, especially as they get older, especially the males. And it's not just the females with the offspring underneath them. So what this means is that if anybody's trying to line breed, or anybody's trying to make any sort of progress, or if anybody's even just wanting to get the lines of stuff that they have seen pictures of over the last, I guess, decade now, and you get a mixed strain, you can get a variable phenotype that isn't what you want, or not what you want for your project, or blah, blah, blah. So this is very hard evidence against the idea that there's sort of a universal expression of these mutations in, at least in scaber. We can just use scaber as a model for a lot of other isopods too. And of course, it's getting even worse with things like the cubaris and nasodillo and troglodyl, etc., because now as whiteouts and oranges and a billion other things are popping up in those different species, the actual species names are being lost, and they're giving them ridiculous strain names without retaining the original species that would give us any indication as to what they actually are in the strain name. Instead of having cubaris species red edge, with the orange mutation, when I denote that, I always put red edge, comma, orange, because it's orange mutation in red edge. There's no other particular strain name. There was no other particular traits bred for in that line. Now people are just putting cubaris species glacier, cubaris species this, nasodillo species this, nasodillo species, cubaris species pink ducky. It's not, the species is not pink ducky. They are rubber duckies that have been bred to be pink. And that's going to create a whole slew of problems. In fact, the worst offender that I've seen so far is people labeling Cubaris marina papaya as Cubaris species papaya. And I know they're doing that just because they want to get extra cash by saying that it's another species and not something that people already have or can already produce. So it's just very scummy all around. And it's just, how is anybody going to have any idea what all these nasodillo and cubaris are when there's a billion whiteouts and they don't have any indication what the parent species was for these strains when they label them with things like blizzard or snow or glacier, and they didn't bother to retain the crappy species name that was already associated with that strain when it's not an identified species. That's the other thing. So like, it's Nasodillo archangelii, Shiro Itsuri, right, is a strain of Dalmatian Nasodilla archangelii, and we have the species name, so it's fine that there's a strain name. But the problem is that things like rubber ducky, blonde ducky, platin tongue song, etc., were functioning as the species name to help us keep track of these lines while nobody knows the identity of all these isopods are. And now that there's been this weird, what's the word I'm looking for here? Not vernacular, maybe weird vernacular push to simplify all of these things. Now we're not going to know what the original species for a lot of these things were if they ever get identified, or the original localities for these things were, without somebody doing some insanely crazy sleuthing on Facebook and forums and a bunch of other stuff. But ultimately, that information gets obfuscated and lost. So it's a big mess, and it's been a big mess for a while. But at least I have this information out there for those who care to read it and utilize it on this varying expression. And the simple explanation for why I think this happens, and I would have to test this by crossing Lucy to, just Spanish oranges, or just anything that has a orange, or I guess maybe a red calico line would be better, because that's the heritability on that is, the phenotype is different than the orange mutation. But to see if such a cross and breeding for the orange, homozygous orange, results in the loss of the eye pigment coloration in the leucistics. Because what that would mean is that basically if the eyes are supposed to be orange or different coloration, then they will, the leucistic will appear to have no eye pigmentation as opposed to having black eyes as in true breeding stock like Lucy. What else was I going to say on this? So, but the reason why I think this is because a lot of lavas have, especially female lavas, have the front half is orange or red. The back half has a lot of the heavier pigmentation. And so, having the Lucy genes crossed into that, these individuals are likely underneath the leucism expressing that sort of dichotomy of coloration, with the front half being more orange, the back half being more black, which would affect the pigmentation in the eyes. But also, come to think of it, I'm pretty sure that even when I had a line, and I need to recreate the line because I lost it over last summer with the lack of husbandry, but it was very easy to recreate because the lava, all those genes are in the lava colony anyways. But I was calling it magma, and it was selective breeding for high orange in lava. And it would still, they would still have some little flecks of the dark slate color on the interior of the body, but they were getting very close to being full-bodied orange, except for the antennae tips, which is interesting because the non-simple recessive orange, full-body orange scaber, as seen in lava, will still have black or slate-colored antenna tips, and I'm pretty sure their eyes are still black or slate-colored. So again, I'd have to do some test crosses to see what exactly is causing this situation. But anyways, so there's some good solid information. And I'm going to look at the chat and see everybody who's beating up on Kai. You guys can beat up on Kai. I bet he can take it. Because he likes Dorcas. He likes the teeny tiny Dorcas. He's giving Parker Blatt a crap, but he likes tiny Dorcas because it was a Dorcas brevis, the one that's only like a couple centimeters. And he'll be like, oh no, they can get to like this much if you raise major male. And then like nobody would raise a major male because I don't even think anybody reliably captive breeds Dorcas brevis for multiple generations in captivity. So that just de facto makes raising any species of Parker Blatta for multiple generations in captivity better than anybody who's in the Dorcas brevis. Although I do like Dorcas titanus. Let's see, blah, blah, blah. A bunch of cat fighting from Kai. Blah, blah, blah. Blah, blah, blah. More blah, blah, blah. Blah, blah, blah. Oh. Is this what I sound like when I talk? Kai, I think you don't enjoy Panchlora because you killed the Panchlora I gave you. I own 60 species of roaches and no Panchlora because apparently you can't keep them going. Parker Blatta don't breed once a year. Fulvescens goes for multiple generations, as well as Pennsylvanica from Alabama, too. I have a good colony, and certain lines of Americana. Although I do have some Caudali from Ellen that I think he sent last year at this time. They're still all subadults, or they're doing stationary molt, so I'm wondering if they need to be cooled or if there's something that I'm not doing right. Why naming species after their color is never a good idea. Yeah, I mean, it's fine. There's plenty of... The thing is, there was a chance for there to be, but there is currently not a culture of preservation and tracking in isopods as there is in roaches. And in roaches, up until maybe 2010 or 11-ish, most of the stuff that we have came from museum and university and other miscellaneous lines within the United States of stuff, and their origins were just kind of, they weren't tracked at that level because universities and whatnot actually didn't want they didn't want the world at large to know that the stock that they were keeping was being distributed into private collectors' hands for X, Y, and Z reasons. And so a lot of the information for some of those strains that came out of laboratories and universities, like Gyna cappuccina, Schultesia lampuritiformis, Gyna bisanulata, Mylina blabrus giganteus, and a bunch of others, don't actually have their collection locality and whatnot data with them, which is unfortunate. But the second that localities of things were, as soon as that we were able to get that information for other things that were being propagated, it became very critical in roach keeping very quickly, and that's in part thanks to folks such as, not to toot my own horn, but myself and Alan and TJ and various other folks who know enough to keep that line information with strains as they're spread around, to avoid strain mixing, to avoid species mixing, to avoid all kinds of stuff. And also because things change too with respect to something that we've had for many years that we thought was X species could end up being Y species. Well, without that additional strain information, that made people say, maybe I should keep this well, low-hanging fruit, but maybe I should keep this panchlora that I got from some random reptile keeper separate from this panchlora that I collected in my yard in Florida. Without that, you can mix species and make hybrids. You can mix species and lose species to culture. You can mix all kinds of stuff together. So the idea of keeping different strains separate, and I will, there is some nuance to this, and the one thing that sticks out in my mind is a difference between Alan and I, which is Alan meticulously labels all of his Parkablata, and I do that too when I go out and collect lines. But my Parkablata fulvescens are actually a cross between Parkablata from they're a cross between fulvescens from Tuscaloosa and Montgomery, and I did that in the hopes that I would increase the genetic diversity and create a strain that was better adapted to my fickle captive conditions, and hopefully somewhere in that mixture be some genes that did not require any sort of diapause or anything of that sort. And it worked because fulvescens are a very reliable feeder for me. I use them very heavily for my amblypygids, among other things, because of this. So, but in that case, I had to make the judgment call, and people can say that I was wrong and say that I was right for that. But I had to make the judgment call that crossing those two localities, for the practical reason of getting the species in captivity, in a culturable form, that was the decision that I made. And so, but again, I don't label them as either of those localities. They're just labeled species because the species is correct. So, and Parkablata notha, Alan started his colony with ones that were crossed between stuff we collected in Cave. I think he caught a male in Cave Creek Canyon, or we only had males from Cave Creek Canyon, and a female somewhere in the Santa Ritas. So it was another very long distance cross. But those, we still have the Parkablata notha strain, and at least I do, and I have to check with Alan. And they have been kind of a pain to deal with, but they seem to be continually going in captivity. And I would hope that over some time, some purifying selection will take place, and they will get easier to culture as the fulvescens did over time. So, but anyways, but Alan, of course, I think he ditched the rest of his notha. Maybe one or two other people have them currently, but I still have the long distance cross strain from Alan of notha. And I did send Alan the only pair of notha that I collected when I was in Cave Creek Canyon last September. So Alan, I think they both matured too. So obviously, if the pure locality stock is going to be easier to work with, I will switch to that. For now, I'll continue maintaining that cross, because if Alan can't get those Cave Creek Canyon ones going and nobody else has them, this could be the only chance to keep a strain of notha going in captivity. So, so yeah, there is some nuance to these things, but again, I don't sell either of them as pure localities when they're not. They're just isolated by species level. They're just distinguished by at the species level on the website because they weren't of hybrid origin. Let me go and catch up in the chat a little bit here because I've been quite chatty tonight. I'm glad we hit some nerves here. Insanely crazy sleuthing. I think I have a new project. Really need people to dedicate to describing Cubaris, especially since it seems a few aren't even true Cubaris. That's why sane people don't keep 200 morphs of scabers. Oh, they will eventually. They will. After having seen Cubaris or Nasodillo species soil and now having white pigeons, I really feel like we kind of peaked on wild Cubaris and Nasodillo a while ago because like tongue songs and a lot of other things are just very similar to each other. Like I could even see how if somebody is doing some bare minimum captive breeding in Southeast Asia and pulling a couple of weird looking individuals and then propagating them and then making them available to the public and calling them a different species. I could see how you could do that within the realm of like 8 to 10 months. There's enough demand for it, and then we'd never know if white pigeon and soil and silver ghost and phantom ghost and colostomy bag and the Iridosoclitus cubaris or nasodillo are actually wild caught species that are actually localities of each other, or if they're just man-made creations within our comprehension that are being sold as different species or something. Look how depressed Alan is. No, you don't understand, Kai. Alan, the Parkablata are bringing Alan immense joy. That's actually why he's not more distressed usually. The Parkablata bring immense joy. Lata is a very beautiful roach, and I am constantly kicking myself for repeatedly failing with them in the past. I do have 1 or 2 sets of them going now from the Alabama trip. But it's actually a very beautiful roach. The females have that nice red and black on the abdomen, and they're good size too. If you like the color scheme of like the African bullet roaches, the Cfneil Silopyga propinqua, I could see how you would appreciate the adult female coloration on Lata. I think having seen them in the wild gives me a better chance at maintaining captive conditions that are conducive to mass producing them. So. Let's go back and look at this. For a while, the iSpot subreddit was full of confusion because PetSmart made Smugbug label their mixed morph P. prognosis as powder blue. Oh boy, Smugbug isn't all bad though. Their autumnal equinox expanses are cool. Just wish they named them differently. I gotta look those up because somebody was talking about those. Expansus. Oh, that might be simple recessive orange, but in Porcellio expansus. And, you know, honestly, at this point, if the ones from Oren don't reproduce for me or don't end up breeding reliably true like the expansus orange that I had back in 2017, you know, it's just such a beautiful isopod. I wish I could get some of that stock again, the OG straight off the boat Porcellio expansus orange stock. The store thought the species name was too unfriendly or something like that. Lotto ticket, maybe, because it implies gambling, which is which is basically what you're doing when you get a colony of the mixed simple recessive Porcellionides. Have you had the same experience as Oren with Gyna cappuccino needing fresh peach slices? No, I have not. It's been 1 week since you looked at me. No, it's been 1 year and 6 months since I was given the opportunity to restart my colony of Gyna cappuccino. I pestered the living hell out of Ty Randall to send me some teeny tiny babies that he had produced. And at that time, I think he may have been the only person in the country with them propagating. And so I paid what was and still is the ridiculous amount for them. And that's, you know, obviously you can all go look at the Roach Crossing pricing for Gyna cappuccino. but I paid it because I wanted the species again, and um, I received them. They were first and second instars, and I set them up, and I power grew them, and I threw them into a setup that's been working for all my other Gyna currently, and I waited, and I got an F1 eventually, and now I think I'm on to F3, and they are doing fantastic, and I'm keeping them the same as all my other Gynas, and they get a handful of dog food, and maybe if I'm feeling generous, some fish flakes and a piece of apple, eggplant, or last feeding session was spaghetti squash. And that's all that they've been getting. And so they do have, well, they did have heat. I actually took them off of heat about a week ago to make room for another enclosure that I wanted to put in that spot. I'm actually getting, hopefully, next week everything goes well, and I'm getting some captive produced Marulinella species tricolor, and TJ will claim his bounty if they arrive safe and sound. But I wanted to try keeping the Marulinella tricolor. I wanted to try keeping them Kyle style in a very bioactive, very rich, very warm enclosure, and see if I could do better than all the people who are killing them, apparently. So, but anyways, so they were on that, and I have a suspicion that they will probably continue to do fine despite the fact that I took them off of heat. And considering at once I lost my Gyna Cappuccino the first time, circa 2019, 2020, and I felt really bad about it. I still had Bisannulata, and I took corrective action immediately to get them going, and I set them up in a 10-gallon. And I think at this time last year I had maybe a couple thousand Gyna Bisannulata, and Bisannulata and Cappuccino have nearly identical husbandry in my experience, and so I set the Cappuccino up as I had the Bisannulata, and again, here we are. They're doing very well, and honestly, the only Gyna that I have that isn't doing well are the yellow Liridas, and I had to move them to a smaller enclosure, and I think they actually didn't appreciate the extra heat I was giving them to try and get them to reproduce. And it's just such a pain because, boy, back in the day, I had a huge colony of the yellows, and they did as well as all my other Gynas are doing right now. And it is kind of sad to see it reduced to one adult male and a bunch of females of subadult and adult status, but just need one good ooth to hatch. That's all I need. I just need one of the females to give birth to a full ooth, and it'll restart the whole colony. And then I will just never sell them until I have another crop of babies popping up in the thousands again, hopefully. So, from what I know, they need individual space to make chambers. I'm not even sure if that's necessary. I grow more suspicious over time with the sensitive Gyna that they're not really all that sensitive whatsoever, that it's more that when the female is forming the ooth, first of all, it seems like some of those, the sensitive Gynas, the females may only reproduce once in their life, maybe only once or maybe twice or something like that. Not many times, though, as evidenced by the fact that the ooths are gigantic. They might even have some of the most eggs per ooth capsule out of any cockroach, because you can just have hundreds, or maybe up to 200, I guess, eggs per individual female's ooth. But while they're forming the ooth, they need a lot of space to create it and to retract it back into their bodies. And I think one of the problems is that in a lot of setups where we kind of cut the headspace for different species in order to stack containers or what have you, that the females just can't get a good location where they can properly form the ooth and pull it back in. And so it gets damaged, and then it gets infected within them, or it gets damaged and it ruins it and they have to abort it, and then that sets them back a reproductive event. And if they only produce once, that could just straight up, then the female dies and they're done for, right? Oh, my poor baby. Actually, it's Victor's birthday today. We went on a nice long walk. We're going to go on another one later. So, but I wonder if that's actually the case, that they just need a lot of space, because Oren had success originally. If I remember what he put on Roach Forum or in private messages, both of his colonies of Capucina and Bisanulata were in 32-ounce containers. And that is a lot of vertical space, but I don't know how much substrate he actually put in there. As individual as macro, oh, you guys are talking about whether or not they need that. Dilophoraptor double-crested thief. What dog food do you use? Dilophoraptor, I have never seen you in the chat before, I don't think, unless you had a different name before, so good to see you. I use I want to say it's Country Squire dog food from Menards. It's very cheap. It's 50 pounds for $20, and that price has remained consistent over the last couple of years as well. And Alex, no, powder oranges should never, ever, ever revert to powder blue. I don't know if you were listening earlier, but I have only had 3 individual Parcelio Scaber Spanish orange revert to wild type, and that was way back in like 2011 or something like that. Those are the only times I've ever had a simple recessive mutation revert to the wild type at any point, and out of the probably like millions of isopods that I've produced over the years. Do you think you'll ever keep Neoblatella detersa? Yes, I have a thriving colony of Neoblatella detersa, but here's the problem with my Neoblatella detersa colony, kind of. So when Alan and Kai went collecting in November, December, when they went to Florida, and I had them recollect some ectobiids that I had lost, I was at, well, I don't think I personally was at a shortage of containers, but I go to the pet store near here and I buy up critter keepers or faunariums and all kinds of supplies. And at the time, I was really setting up lots of new cultures. And I actually didn't have enough of these faunariums to make an individual container for Neoblatella and Latiblatella. So since they have kind of similar care needs, I put the 2 of them together and I created a mixed culture of Neoblatella and Latiblatella. And they're pretty easy to tell apart, but the problem is that when I was trying to remove them each into their own bins, it's very difficult because of their behavior to make sure that I was only grabbing Neoblatella or only Latiblatella when I was moving them, because they jump up my fingers, or I'd go to scoop one up and I'd pick up 3 and then I have to knock the other ones off my fingers or whatever. So I did finally get to, because I finally bought enough more containers to make more enclosures. I did end up making strictly Neoblatella and a strictly Latiblatella container, and I think I finished that about a month ago-ish. So any day now, they should start to establish as the adults that I moved into them start, the ooths start hatching. So I do have Neoblatella detersa, and I should have them into the future as well. So I should, well, I could put available on it, but again, I don't want to have to pick the quantities that I have available on the site out for orders out of the mixed culture, because that will take a long time, because again, I don't want to get Latiblatella in there for sales when people are expecting just pure Neoblatella to be in there. So it'll probably be, I don't know, a couple weeks or months as they infest the new enclosure, and then I'll be able to take the status there. But I do have the cultures going; they're doing well too. All right, I can just say Dilo. That's fine. Let me pull up the dog food country. I might go get some orange juice too. Is this the one I use? It's not the high protein. Because again, who was telling me about protein the other day? I don't want to hear another word about protein in roaches or isopods. People saying they're protein hungry. You know, let me get my orange juice before I go on a rant. This is the rant Roach Crossing episode. Dilo, here's the dog food that I use. Seems with the rebate, which I never do because I'm apathetic to it, gets you down to about $17. So I'm gonna go grab some orange juice and be right back. Okay, and I can lift the shade because the sun is behind the clouds. Boy, do I look pasty even though I'm not pasty. Well. Apple juice is good too, but there's too much added sugar in a lot of it. So I like the orange juice because you don't have to add any cane sugar or high fructose corn syrup. Anyways, but yeah, marketing stuff is and just looking up at Dilo's thing here about orange basically being albino, powdered blues, wherever. The marketing with pet stores is, now that roaches are going they've been mainstream for a while, but they're things like that and isopods are mainstream now. Is they don't want to compete with the other people, and so changing a common name to something helps you sell stuff better. And so having an approachable or fun common name really helps you sell stuff better. One example is, you got all the beta buddies line stuff for betas when you can just buy all that stuff online. But also they're hissing roaches. If you've ever picked up a container of hissing roaches for sale at a Petco or PetSmart or whatever, they call them giant islander roaches. They call them giant islander roaches. No mention of hissing. The main thing that drew a lot of people to them in the first place and made them popular in the first place, giant islander roaches. And that's to avoid competition with all the people that sell them online as hisser roaches, expecting somebody to see a giant islander roach, purchase it, and then go home and make a forum post and be like, I got a giant islander roach. Is this a hissing kind? They've already spent their money. So anyways, what was I going to rant on protein? Aren't large Porcellio and A. klugii protein hungry, or is it more of its availability? I have had pretty much every isopod that I own go to nibble on my fingers at one point or another, and that's not even when there's food residue or something on there. Pretty much all of them will go for easy meals, especially if they're hungry. They will eat each other. There are some of the species that are considered universally safe, like Porcellinides pronosus. I know more people who have had roaches that have been chomped on alive by Porcellinides pronosus than any other isopod that I have heard of. And it's all about the husbandry and what you put in the enclosure. If they're starving and they're desiccated, they're going to go for whatever they can to fill those needs. So if your husbandry is crappy, then your isopods will be hungry. If you have a nice, rich, biologically active enclosure with plenty of microbially active decomposing leaf litter and rotting wood or whatever the species prefers, you will not have any of those issues, which is why I keep a colony of Porcellio scaber with a lot of my millipedes, including my Centrobolus. And I remember saying the customer being like, "Oh, don't the isopods like eat the millipedes?" And I sent them a picture of my Centrobolus enclosure, which had just a pile of Porcellio scaber just on top of another pile of millipedes. And they were fine because the smaller millipedes live deeper in the substrate, just like the smaller Porcellio scaber do. And also it was just all of it was pure rotting wood and leaf litter. And this also wasn't a freshly set up enclosure. This was maybe 8 to 9 months after the enclosure was set up. So it was being actively eaten. And there was all the organisms involved, their digestive systems, the microflora within them must have been doing very well too, so they didn't have to seek out unreliable or irregular sources for different nutrients and whatnot. Yes, and the Tennyson, and as long as you have proper food sources, if your husbandry is good, you can keep a lot of different things together. In my living room vivarium, which currently the peppered roach colony is starting to swell because I set them up and I said to myself, I really like this roach species. I want my colony to finally do well again, so I put them into a large enclosure and babied them, and they're doing exceptional now. And I also have had various isopods that have been mixed in there with them over time. And the ratios of what does well and what doesn't depends on some of the inputs and the density of the roaches and density of the other isopods. But you can fairly reliably keep most single species enclosures of roaches with a single species of isopod at least without any problems. I keep my klugii. I keep them with Montenegros are with Gronfodorina, the black tiger, or whatever. TG and I probably had this discussion on another live stream where TG and I were trying to figure out what the exact situation is with the important the Grandadiri black. They are looking more and more likely to be Portentosa, but I'm hesitant to label them that because then people mix them with pure Portentosa, and we have no idea where they came from. So, anyways. But I keep my klugii with those. I've kept them with ivory millipedes. I've kept them with stick bugs of various sorts. You can do a lot of communal enclosures. It's like you can't expect colonies of things to do well if you throw them in a small shoebox with limited resources, limited ventilation, and not proper microbiota. You just people throw things in a shoebox and wonder why they die, and it's like you created a death chamber basically where it's stagnant, and only the things that are really well adapted to these conditions, or if the colony you're throwing in is large enough, you get good allele effect going, but then eventually they'll exhaust the resources and the colony will crash. So some Vulgarian Nazatum in my communal invert enclosure, and I have not seen any issues with any of the inverts in there. Yeah, and that's another thing is making sure there's enough, there's a good diversity of microclimates for different things to inhabit as needed. And, again, small enclosures can work, as you can see from these small enclosures. And I keep all my isopods basically in the same size enclosures other than the ones that are living with roaches. And it's all about, accommodating knowing the species enough or having the information on hand about the species or strains that you're working with enough to set up for success and then maintain that equilibrium as long as possible. Like a really good example that I can speak very confidently on is I keep Porcellio nicholsi, a species that likes it very dry, with Marulinella species Vietnam, a species that likes it very moist. And the reason that works extremely well is because there is enough of a vertical and horizontal humidity gradient that the Marulinella can live in the lower layers and the nicholsi can live in the upper drier layers. And there's just a bit of there's a bit of mixture of each species into the other species territory, basically, in that situation. So, but I do find the younger nicholsi like it a little bit moister, so they'll be down in the substrate of the Marulinella. And sometimes I will find big Marulinellas, maybe males that have been pushed out of the area, or just for whatever reason, it's humid enough, they'll be up hanging out with the nicholsi. And there's a couple hundred of both in that enclosure. And I consider that a very good success. And it's been going that way for about a year and a half now. So, and of course, I top off leaf litter and whatnot and feed and water regularly. So I feel kind of like a broken record with some of this stuff because I know I've talked about it, especially on Russ's live streams. But good bug cultures, healthy bug cultures can go without maintenance for a long time with an asterisk. Some species, like I would not leave the Hemipterosaura to their own Vittata to their own devices for over a month without some sort of inputs unless I really, really, really prepared in some way, probably with a wick water or some other things. Or Latibotella just because they're just a finicky species to begin with. But things like Leucia hermetica and Glabrus and Versatria and Eurycotis and Gronfodorina and Ailuropida and a lot of those things, my cultures go a month that's been the average nowadays. And if anybody's been paying attention to me doing the live streams in between when I do blog posts or other times that I've commented on, or if you've inquired about how often I take care of my roaches, it used to be every 10 to 14 days when I was younger was when is how frequently I would take care of roaches. Now it's like every 2 to 5 weeks, depending on how busy I am. And most things are just fine, and I have learned which things are not fine, and I do put more effort into taking care of them, which is why a lot of the stuff is up here, and some of the stuff I just really like, so I want to make sure it does really well. But yeah, I mean, there's people. All my isopods. Oh boy, I brought this up already. All my isopods over the summer last year got no care, including no watering. There was no watering going into any of the cultures. And when did I even start doing this year's isopod overhaul? Maybe in December or something like that. But I can't remember if it was 5 or 7 months that some of those cultures went without any food input or any water input. And I was able to salvage most of those colonies. I mean, some of them were there were 3 individuals, and some of them it was like, did I take care of this yesterday? Why are there so many? Why are they breeding? Especially Armadillidium CF Espanoli, the marbleized or whatever. Those, I think I opened up the bin and there was like substantially more than I had left in there when I had last checked on them many months prior. So I'm about to rant about protein. Yeah, I am, Alex. Eventually, I get to that. I think my blood has cooled off a little bit about the protein rant. It would be nice if you could take pictures of your enclosures and maybe also list the food and parameters and make a blog post about it. Yes. So, my intention this upcoming winter, and Chris Snyder is not here right now, but Chris and I have been talking about some major changes to the website, including what I'm thinking is long overdue, perhaps, is a major overhaul to a sort of final form, which has very easily searchable information about all the species and strains that I have in terms of size and husbandry, and a lot of information that's already on the site, but more easily searchable and whatnot, which will take a long time and a lot of effort and technological know-how to implement, but is on the horizon, perhaps after I move. But I do want to, at least by this time next year, have a new iteration of the cockroach care guide, which would have a lot of the information. And I would also like to do some videos of me putting together enclosures of things. But the problem is the amount of time and effort that it takes for me to record a video. Sometimes when I'm putting together enclosures, or perhaps 70% of the time, there is some urgency to me getting something done and getting things moved. And so I don't have the husbandry wiring says, no, don't record this. You need to get this done so that these things don't die, or so that you have a good colony in 8 months or something like that. So don't sit to think down and do that. So if I did anything, I would probably put together a mock enclosure or put together an enclosure for isopods or something to just have on hand, because I'm constantly splitting new cultures and stuff like that. So Tennyson says, Roach is going out of the moisture while the Blapstinus larvae, springtails, and isopods tend to stay on the moist side. I've seen this behavior for a while and I'm now comfortable enough to check on the enclosure monthly other than feeding. Still a little paranoid, but it's fine. Let's see, where is it going to this? Okay, moving past the Roach helping. I hear a boy who wants to come in, so I'm going to go let him in because it's his birthday and he can co-star on the stream for a bit. Hello. Hello. Come on, it's your birthday. See how you go ask you. Come on. Maybe not. Okay, you know where I am. Let's see. Do Halloween hissers sing to each other? I've been doing a lot of hissing. Yes, each hisser species has a unique call, and they also have different calls within the species to signify antagonistic behavior and courtship. And actually, I think I sent many years ago. I sent some series of roaches off to somebody who was doing research on that. I think they needed a Leptorida davidii for that research because they couldn't find them anywhere else. Maybe do a whole week of blog posts detailing husbandry with some of your inverts. Yes, and I think for that I would want to make a fresh culture of something or make a fresh enclosure, document that process as well as moving things and whatnot, and then also have some of just the day in day out. Because actually today, Christian and I did just regular husbandry on a lot of stuff, and a month's worth of husbandry was basically I eyeball it and I see if I need to add some more bark pieces or refresh some of the mulch substrate. I throw in a handful of dog food. I throw in a piece of eggplant, and then I spray the whole thing down and I eyeball it and think, is this going to give me any problems next month? And then I usually tell myself no. And if I do, I'll pull the container off to the side and I'll do more intensive work on it over the next week, 2 weeks, depending on how busy I am. But anyways, yeah, mostly monthly maintenance, or each maintenance session usually only takes a couple seconds per bin, but there's so many per tank, but there's so many of them. What are you doing? Oh, he's a good boy. He's a good boy. He's a 1-year-old boy. What? You can come in here anytime you want. Come on, come here. Come here. No? Okay. Being a little brat. The 1-year-old doggy time. You want more people keeping isopods and critter keepers? Well, maybe if they did, they wouldn't fail with so much stuff. Why do you put Thuria as intermediate? I've seen a lot of people say they are easy, but some people seem to disagree. Let me go and see if. What do you want? Are you going to come in here? Come here. Good boy. All right. The stream has been hijacked by the dog, who is just off screen right here. There you go. Here's your boy. He's a good boy. He's a good boy. Yes, he's needy though. Watch if I stop paying attention to him. Oh, poor baby. Oh, on a quick aside, I got his Embark, his genetic testing kit came in today. So his getting him genetically profiled to see if he is fit to be breeding stock, and considering there's literally no history genetically of any problems in his family, he's probably good to go, and he'll just need to get his elbows and hips and stuff checked out and then do some livestock working trials, and then the studying begins in 8 months. Tennyson. Let me get another sip. Well, this is going to be a good length live stream. Still getting used to the beard. I put them as that because, and I'm going to look this up on my terms because I know it's on here somewhere. No, it's on the using the site page. How many people here actually have read my terms and conditions in my using the site page? I wonder that, because there's a lot of information on both of those pages. In fact, on the using the site page, which again I would think people would use, there is the little thing that so many people overlook, and I even put something so that people wouldn't overlook it. But each page has the detailed species stats, and I think people see the little arrow for the drop down, and they think that the little flavor text blurb that's underneath that is the detailed species stats. But if you click on that, I have all this. I have adult size, climbing abilities, flying abilities, mode of birth, care level, temperature requirements, air humidity, substrate humidity, favorite foods, locality. All of this information is on that drop down. It's just not in the little flavor text. And so for the future site, I think that information is going to be more in your face presented somewhere. I just put it there so that people won't get overwhelmed with the hard numbers. But it seems a lot of people might actually want that in this situation. So Tennyson, to explain why Thuria are intermediate, not easy. Reading off of my care level blurb. Easy species are very forgiving and tolerate many different conditions or are very low maintenance. Little animal husbandry experience is needed for these species. Some examples are common mealworms or dubia roaches. Intermediate species are still quite easy to keep, but may have one or more special requirements to keep them healthy or breeding. Some examples are pie dish beetles and domino roaches. So in the case of the domino roaches, there is still the pervasive idea in roach keeping, at least it's very strong for feeder keeping, but I get asked this and told this enough that I have to put in there that you can just keep roaches on egg cartons. And you can do that with some stuff. And some people, because of their humidity and other conditions in their enclosure setup, a lot of things do just fine that way. But you can't just throw Pseudoglomerus magnifica into a tank with some egg cartons and expect them to thrive without complex, very involved invertebrate experience micromanagement of humidity and other factors. Same with Thorea, people will ask, "Oh, can I just keep it on egg cartons like Dubai roaches, And no, you cannot. You have to have a substrate there to retain humidity and for them to hide in and feel comfortable in, and blah 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 blah. So something as little as just needing a substrate can bump a species on my ranking from being easy to being intermediate. Because something like lobster roaches, you can throw into an enclosure with no substrate and they'll do fine, or pallets, or dubias, or a handful of other things. So that's why domino roaches are ranked as intermediate, is because there's just that extra little step above the average assumption about keeping different roaches. Just that extra little step above that. Let me go up here and see. Dilo says, do you have any of the pill roaches, E. Chopardi, and decent beginner colony availability numbers? Yes, I have plenty of E. Chopardi, and for pill roaches, so Pseudoglomerus aren't a pill or roly-poly roach. The Pseudoglomerus fake ball, they're related to the Perispherus, which can roll into a ball, but they cannot, and so they're genus Pseudoglomerus. I have had historic success with Perispherus pygmaeus, and lately, anytime that I've tried to try them again, I have not had any success. So I don't have any Perispherus pygmaeus available, and likely will not for some time until somebody like TJ gets a good colony going and has some to spare. And ideally, if anybody ever has them for sale, I want to buy like 100 to get the Lee effect right off the bat. However, TJ did send me some Perispherus punctatus, which seem to be a lot hardier than pygmaeus right now. So if my colony keeps doing as well as it has been, or rather, if the individuals I have do well enough to establish a colony, I could see Perispherus punctatus becoming quite popular, because it would be a very easy to keep pill roach. So, why dog food and not fish flakes? Fish flakes are expensive. Quality fish flakes are expensive, and dog food is not. Dog food is also low protein. I gotta get on my protein rant. I gotta do that after I finish reading through the comments here. Dog food is lower protein. It's more cost effective, especially when you're feeding colonies of large roaches. I have primarily been feeding my isopod colonies with fish flakes, the individual colonies of things, because it seems to have some, the ingredients are less grainy, and thus, even if my husbandry conditions within the bin are very bad, such as if I've overwatered or if it's understocked or something like that, the fish flakes are more likely to mold or be infested with springtails than to be overtaken by grain mites, as what happens with dog food. So, but with the dog food and roach bins, where I offer it to them, it stays dry enough that it doesn't. Excuse you. It stays dry enough that it doesn't attract grain mites, or gets eaten quickly enough that it doesn't have that. That just isn't even a factor in the equation. So, and the roaches quite like it too. That's another thing, is I tried a lot of different dog foods over the years, including going down the protein route, and these low quality kibbles just seem to be perfect in tandem with the fruits and vegetables that I offer. Do you want to come here? I'm not coming to you. I'm in a live stream. What is that? Is that my underwear? Victor! Why did you do that, huh? Why'd you grab my tiny whities? Huh? You silly boy! He can be a little bastard sometime. All right, he wants you to do the protein rant. Oh, hi TJ, good to see you. Let me see. Let me go up here. Dog food, blah blah blah. Trying to see if I missed anything. I want to make sure everybody gets their stuff read and anything that needs to be answered gets. Nofita, the only species that eats potato. Yeah, that potato is kind of a meme that like a lot of universities and stuff seem to offer their various captive invertebrates potatoes, but like raw potato isn't like good, and if it gets green, it starts to produce the solanine or whatever, and then it becomes toxic to everything. So I don't know where that sort of caught on, you know. Moist paper towel corner. Then why is Pseudomonas septentrional easy? That's a good question. Maybe that's one that I had overlooked and need to. Maybe I've kept them without substrate at some point. But you are right in that may need to be bumped to an intermediate species. But I would have to think on what Pascal's logic was putting Pseudomops down as easy. That is a good point that I may have to update. And this is another reason why I'd like to move, or when I move the site over to a modern format, which will be more aesthetic and blah blah blah. I would be updating all this information with the experiences that I've had. I love how TJ is a natural check to Kai's savageness. It's truly beautiful, I must say. I remember hearing dog food sterilizes the roach. There was, before I go on the protein rant, which I've been waiting on doing, there was a guy on Roach Forum who went off on TJ. You and him had an exchange, is what I read this at some point. Some guy went off on chick feed on the Roach Forum, and he seemed like he had there was something substantial to what he was saying, like something valid about some of what he was saying, but then he just sort of spun to this absolute craziness about like he was like, oh, they've been adding this to chick feed since 1993. They passed this legislation, and now they all treat it with this thing, and you'll notice this and this. And I raised the dubias on this, and they all died. And then I tried it on this, and I added this, and they all lived. And I don't believe when anybody says the dubias, they don't become adults or something. It was just such a weird. It's a very specific type of person that has these sort of opinions and stuff on things. It's very interesting. But it was I don't care enough because I have a strategy that works for maintaining my roaches and stuff. But like, is it for me to look into see, oh, is this guy crazy, or was he really onto something about them adding something to the feed that is pesticidal or something like that? But here we go. TJ, yeah, that dude on the forum was kind of nuts. Dog food and unmedicated chick feed are perfectly fine for roaches and have been used for them forever with good results. Yeah, so I don't doubt that there could be something in the process that is used that could affect the vigor of roaches, perhaps in dog food and things like that. I don't doubt that that's a possibility, but again, this guy I read that and I was like, yeah, I can understand. And then he just went off the rails, and I just found very hard to reaffirm the blossoming belief in whatever he was talking about before he started talking like a crazy person. So, um, but yeah, the protein rant. Basically, with roaches, their entire physiology for most roaches is based around extracting nitrogen from nitrogen poor sources. And nitrogen is one of the primary molecular components of proteins. So it is possible to very easily give your roaches gout by feeding them a protein heavy diet. And many behaviors that were blamed on protein hungriness. And this is, here we go. Now we're getting really into what I want to talk about. When I got into roaches, the protein hungry orange heads, protein hungry Eublaberus posticus. It was like everywhere. Oh, they bite the wings because they're protein hungry. It's like, what? What? And then, so I would feed them everything, and they did well for me as Eublaberus tend to do and whatnot. Although lately I haven't been keeping them moist enough. We'll get into that, anyways. And I would still have all these problems with the wing biting and whatnot. I was like, I'm feeding them protein. What am I doing wrong? And then one year, I kept a big colony of orange heads in my parents' garage in a big bin. And it was over the summer, and it got blistering hot in there, and it got super duper humid, and I would forget to feed them all the time and all this stuff. And I watched over the months that they were all maturing with good wings. The vast majority of them had good quality wings, beautiful wings. And I thought, like, I'm like neglecting these. Why is this happening? And then I tied it together that this wing biting or this resulting phenotype that causes this wing tattering is not due to them biting or chewing on each other's wings. It's due to, or it's not due to a lack of protein, it's due to a lack of moisture. There are a handful of pictures of Eublaberus posticus in the wild, and there's some notes on them in the cockroaches ecology book on their habitat, and they are usually found in very moist, almost wet parts of caves. In fact, let me see if I can pull up a picture of this. So moisture is, and the other thing validating this, and giving some more context, is that in the same cave you can find Eublaberus posticus and Eublaberus distanti. The distanti stay in the drier parts of the caves, and the posticus stay in the moister parts of the caves. So there's a picture that shows them just on absolutely gross substrate somewhere in like a bat cave, and I want to find it so I can link it here because it looks really grody. But it's like this is their habitat is this sludge at the bottom of bat caves, this moist habitat, and that's why they did so well in that enclosure over the summer. Gotta be able to find this picture. Cockroach bat cave. Pictures of bat cave cockroaches. Here we go. Here we go. Perfect. Copy image link. Go to page. Grab this. This is a beautiful image. Oh, and I'll catch up with some of the people who popped up here. So this is a picture of Eublaberus posticus in the wild. Look at how disgusting this cave looks. It just looks like it's covered in like poopy sludge, which it is, literally covered in poopy sludge. And you can also see, even more interestingly, even in the environment that they are adapted to thrive in, that these posticus have, some of them have tattered wings. You can see that very, very clearly here that a good portion of them, maybe as much as 40% or 50%, have these altered wings for whatever reason in this bat cave habitat. So this is a very natural thing for them to have. And again, it seems to be off-put by keeping them more humid. So that was the thing. I don't know where they got. These people seem to think that protein is some extremely ridiculously abundant resource in nature that that everything is competing specifically for it, and that everything is hardwired to just go for it without any nuance or thought that, especially in tropical places and in leaf litter environments, pure protein is not a common, it is not a common and very easily accessible thing. There are mushrooms and whatnot. There are mycelium that colonizes things and converts nitrogen in that to a more accessible form to organisms. But detritivores, things that feed on decomposing organic matter, unless they are specialists on things like carcasses and whatnot, are living in conditions that are not necessarily nitrogen or protein. They're not nitrogen rich, and they have to have different physiologies and means of acquiring the nitrogen that they do need for their bodies. And there are different ways for doing this. In millipedes, a friend of mine, Mike Underwood, he does a lot of publication crawling. It was read that millipedes are basically bioreactors, and when they eat decomposing material, the concentration of active bacteria inside their body cavity, inside their digestive tract, is almost 3 times higher than the concentration of bacteria in the ambient leaf litter environment around them. So and it's not a specific bacteria in that situation. They basically take in the endemic bacteria of wherever they're feeding and create a very choice microenvironment within their bodies that allows the bacteria to decompose the wood and leaf litter and whatnot that they consume. And the millipedes then are either digesting the byproduct of that, or in a manner similar to some grazing animals like cows, they're physically digesting the bacteria, because the bacteria have the nitrogen in them, not the broken down cellulose and whatnot that they have been consuming. So that's the protein rant with roaches, but even more so, roaches, again, I was going into how they have complex physiology to preserve nitrogen. They are extremely good at preserving nitrogen, and if you feed them too much protein, they will get gout and die, especially if they are a species or a taxon that does not have an adaptation to get rid of extra protein in their system or extra nitrogen. So blabberids do not have a means of getting rid of extra protein in their body system. You can kill them with gout very easily. Blattids may or may not, I'd have to do more research. Ectobiids do have a way of getting rid of extra nitrogen, and it's thought that because they can do this, that they're kind of passively storing it in their living space for consumption later. And so if you feed Parcoblatta, Paratemnopteryx, I'm trying to think of another species I've seen this happen with, Annelacta, too much protein, their poops will come out white, little nitrogen pellets. Think like bird poop white, and that's because their body is actually ridding itself of the extra nitrogen because it can't handle it. They will get gout, and they, for some reason, have an adaptation to get rid of this. But this whole nitrogen recycling that roaches do is because of the bladder-bacterium that they have a very intimate mutualistic relationship with, to the point where roaches have very specialized organs within their body that house the bladder-bacterium, which is what allows them to recycle the nitrogen within their bodies. So it means that they can bioaccumulate nitrogen from extremely poor sources because they are able to basically recycle a lot of the nitrogen that they do use and consume within their bodies, which is incredible, which is why roaches are so durable and are able to feed on such poor quality things and persist, is because of this mutualism with the bladder-bacterium endosymbionts. And again, this is based around preserving nitrogen within the body. So a lot of them do not need to consume extra nitrogen or extra protein. Again, nitrogen equals protein in this discussion, basically, because they're able to recycle a lot of it within their body. They don't need to seek out extra proteinaceous sources. Now, isopods, it's a different story. There's been a lot of literature on this, and this is why Kai got a snide comment in about my reputation within the isopod community because of this protein situation. Isopods basically live in places that are very biologically bioactive places that have a lot of decomposing leaf litter, decomposing wood, etc., depending on the species. And they also are primarily active in the wild when there's higher humidity, which corresponds with greater fungal growth, greater bacterial growth, etc. And so they are able to select for food items that are actively colonized, actively growing or propagating bacteria and or fungi to acquire their nitrogen, to basically get the nitrogen that they need. They are very good at selecting for food sources that are that have bacteria that are making nitrogen very bioavailable to larger organisms. And so this is why isopods like things such as actively rotting leaves and poop. They absolutely love to eat fresh poop because, especially if it's omnivore, especially if it's or avian poop, because of the availability of the nitrogen in the form of the bacteria that have colonized the feces. And so this whole notion of isopods being uh, crazy, certain species of isopods being crazy protein-seeking predatory creatures is just absolutely ridiculous, because it is disadvantageous for them to use energy to hunt things and to develop that, at least isopods as we currently have them, given millions of years of adaptation and whatnot. Who knows what pressures and what things could come out of them. But this is not to say that isopod species do not have different dietary needs, but what it is saying is that the reputation that a lot of them have gained, especially when the metric is, oh, Porcellariides are so good and they won't eat this and they're so not protein hungry, and oh, Porcellarioscape are so protein hungry and blah blah. Those species are very similar in their physiologies and in what has already been studied on their biology, that these are false equivalence statements, or they're not, whatever the opposite of that is, that they are seeing something that is the result of poor husbandry or poor food availability, and they are then blaming it on something that is innate to that creature when it's innate to all of those isopods, that they will go after things. Again, I have a video somewhere. If Derek, I have to check my messages with Derek, where I had an Armadillidium maculatum crawl onto my finger and start eating my skin. So do I run on Facebook and do I tell everybody not to keep Armadillidium maculatum with anything because they're protein hungry because it attacked me? Or do I say, maybe I should feed them more, or maybe I should replace the leaf litter, or maybe I should make the conditions in this bin more conducive to the species? It's like saying if you've got a bull penned up in a cage and got its balls in a vice, and then you let it out of the cage and it starts goring people, and you start saying, bulls are vicious creatures, look at what this thing does, versus saying, like, maybe I shouldn't have locked it up in a cage and put a vice on its balls and abused it before letting out of this cage. Maybe I should have gotten out of the way before I ended up releasing this pissed off creature. So that's my protein rant. And people can say that I'm crazy or that I'm dumb or something like that, but I will say that my large number of cultured isopod species and strains list, as well as the complex enclosures that I keep multiple species of roaches, millipedes, and isopods in long term, should speak for itself in this situation. So rant over, and I actually got a lot of people here. Maybe I should do angry rants more often. Like, oh, that roach crossing guy, he actually has strong opinions on things. He's not so happy-go-lucky after all or something. So let me scroll up and see. Hi, Russ. Also, good to see you here. Hoping to see some young Vanonis. The adults I got from you, Kyle, seem to be doing well and providing lots of fruit flies and fish food pellets, and there are loads of springtails in there. Good, good. Yeah, you should. You may see, and I can't put my finger on this. I really should think about it more closely. But say I it really seems like you can go from having like teeny tiny babies to full grown adults in a very short period of time. And I should really separate some teeny tiny babies and watch them grow to adulthood. But you can sometimes, you'll miss that you have babies and you'll just see more adults. And because if you already have a good number of adults in the enclosure, you'll just sort of be like, huh, that kind of looks like more. So just keep an eye out for that. TJ says, immatures do fine with the adults. It's any new adults emerging that are instantly being put down by the old adults for some reason. So I'm setting up satellite cultures in my ice spa bins picture that I included. Wild caught Venonius to stop killing any of the offspring that mature. TJ, actually, I have you sent me some Ornatus from Ocala, because my culture just never took off from the ones that or maybe it produced just a couple from the ones that Kai and Ellen brought me. Sinusoides are doing great though. I have plenty of those, as well as I did get some Sei in from Oklahoma today actually, and they seem to have darker legs than the Eastern Sei. So I got to set those up still, and I'm really hoping to get a colony of those going. And I also got some Sei in from St. Landry Parish in Louisiana. I've got to debate which ones are going to get full-size enclosures and which ones will get smaller enclosures. But I think the Ornatus that you sent me, TJ, are all still immature-ish. I have to check on them. They're up there. That container with the orange label is the Ornatus from Ocala. But there's constantly fruit flies crawling around in there, and I found or feel like that discourages them from doing any sort of cannibalism and whatnot. So feed with less protein would be best for blackbirds. Now, Tennyson, I will touch on that. There are studies that roaches are very in tune with what their current requirements of stuff is. And so they will select from a variety of things to get the micronutrients that they need at that moment. So a gravid female will select things differently from a freshly matured adult, which is different from a newborn, different from a subadult, etc. So by giving a couple options for them to eat at any given time that they can easily pick between, you're setting yourself up for success, because they may want more carbs, they may want more sugars, they may want more protein at the time, for whatever reason. So, and Dr. Appel and I had discussed this a bit also. Happy roaches, I think, happy warm roaches usually go for more sugary things, and cold depressed roaches just don't eat anything at all, or they go for carbohydrates or something like that. I'd have to talk to him again, but there are different preferences within the species life cycle of different roaches, so it's best to offer a good diversity of things. So Shane, Kyle, your brain is a treasure. Oh, thank you so much. Do you do lectures? This kind of counts as a lecture, I suppose, so I guess I have to answer that as yes. But I haven't really done any invertebrate lectures. I was supposed to give one in Auburn when I went down there, but unfortunately it could not be arranged. Instead, I got an awesome tour of their roach facilities, and then Alan, Will, and I went collecting and stuff. But I do give lectures on native plant gardening, just gardening in general as well, since I have a very accessible venue for that. Actually, I have a talk coming up in June I have to get a presentation ready for. Dilo, is too much nitrogen bad? Same with protein. It sounded like ammonia was nitrogen greater than protein. Proteins are made primarily of nitrogen. So when I say nitrogen, nitrogen is the building block of proteins. So it's just easier to refer to in an ecological sense as nitrogen, because usually when you're discussing things in an ecological sense, you're talking about raw chemical inputs to systems. And so when you talk about the bottom of the food chain, you're talking about the primary producers like the plants. So you have a very nitrogen-rich environment, one like a grassland, where there's a lot of fertility in the soil and whatnot. There's a lot of grazing going on, because the photosynthesis that's powered by the you feel like a dummy in a second. By the chlorophyll, which is nitrogen is needed for a lot of plant processes and enzymes to actually gain the biomass made of carbon. you're talking about nitrogen, it's a limiting factor in a lot of ecosystems. So nitrogen poor ecosystem, at least with respect to the leaf litter or substrate, would be something like a tropical rainforest, which is surprisingly very, very, very low in nitrogen for a couple of reasons. Because one, at least in South America, because of all the rain, or I guess anywhere where there's lots of rain, the rain washes the soluble nutrients into the bodies of water, which is why things like the Amazon are so lush. The Amazon River is so lush is because it's getting all the nitrogen washed in from the surrounding areas by the constant rains at certain times of the year. And two, because any nitrogen that's made available in the soil or in the leaf litter and substrate is readily taken up by plants, such as any of the larger trees or any of the millions of different plants that live in the tropics. So those environments tend to be very nitrogen poor. In fact, this is kind of interesting. If you watch a show like Naked and Afraid, and you watch when they send them to somewhere like just this kind of apples to orange. It's supposed to be apples to oranges, but just kind of they do a lot of episodes in South America and then a lot in parts of Africa. But if you compare when they drop people in the middle of nowhere in the rainforest versus when they drop them in the middle of nowhere in parts of sub-Saharan Africa, grassland Africa, they have a lot of problems finding food in the rainforest. You think that it's this lush tropical paradise, but most of the plants have chemical defenses. There's not a lot of large animals around for things, a lot of huge trees and stuff like that, but there's not a lot of plants. There's not even necessarily a lot of fruit available because all the birds and monkeys are up in the trees eating it there. So they get dropped in the rainforest. A lot of they end up fishing or they end up eating like the odd snake here and there, something like that. But those ecosystems are not very nitrogen rich. There's not a lot of available nitrogen. You see them somewhere in a grassland, they're hunting guinea fowl and impalas, and there's fruits and shoots and all kinds of stuff for them, and that's because the sunlight in combination with the richness and the availability of nitrogen in those ecosystems makes it a lot easier for them to find food. Just a little aside, I like watching Naked and Afraid with my parents because I always wait for weird or cool bugs or reptiles to pop up. So I'm like, oh, so that's where that lives. Little 5-second shots they do between the actual show and whatnot. Let's see. Nitrogen protein. Okay. Might need to change the dog food you're using, Kyle. What I have is fairly protein hungry or fairly protein dense. Yeah. Cat food. Here's the thing. If you offer cat food, dog food, a fruit or a vegetable to your roaches, you can find out very quickly which they prefer, and then over time adjust what you give to your colony under those conditions. You can't go wrong with offering a buffet and figuring out what they like to select most frequently off of the buffet. So you might have, your colony may, for whatever reason, need lots of protein. You might have a lot of females that are producing egg cases or things like that. They will select for the protein in those situations, but you may find that that's too rich for them and they all go for the dog food instead. So, but in general, they're not going to want to eat, in general, roaches need far less protein just de facto than most other invertebrates do, including other detritivores. Shane says, Dilo, protein is a chain of smaller acids that have nitrogen as their foundation. Eventually, the proteins get broken down into nitrogenous compounds. Thank you, Shane, for going a little bit deeper into the chemistry, because I have a fair background in chemistry, but mostly it's at the conceptual level with ecological concepts and the availability of things in very bite-sized terms, such as available nitrogen versus the backgrounds of this, and this is how all these things very complexly interlay. So Alex says, do you think the type of wood used to make flake soil would affect isopod roach performance? Yes, and some of that's because of the innate phytochemicals that might be in the wood or aromatic oils and whatnot, but once it breaks down, I think it was Chris Snyder who brought this up or maybe he brought up in a message to me, but he was looking at how different wood and hay and stuff gets broken down by different fungi and looking at the end results in categories of organic compounds. He was saying like, oh, this ends up being like 70% carbohydrates and 20% sugars and something like that. I have to look up his email. So different things do get broken down different ways. And I have a big compost in the back that I dump all the old mouse refuse and guinea pig refuse and yard refuse and all kinds of stuff into. It becomes a big, really good, rich soil and whatnot over time. But I can grab it at certain stages and use it for different invertebrates, which is what I wanted to do for the Marulinellas. So I grabbed some and I mildly sterilized it, then I recolonized it with by squeezing some water from hydrated compost from the bin into that to sort of get the bacteria and fungi out of it without getting the macroinvertebrates and microinvertebrates. But anyways, it's pine, but it's got a lot of or other organic matter in it from being mixed with rabbit poop and mouse poop and stuff like that. And it's broken down enough that it's not as they call it, hot. I guess that's when there's a certain cast of bacteria and other things that are colonizing it. It's not ideal for some stuff, or not ideal for certain applications. So, but yeah, I use pine and I feed a lot of stuff. I use pine that way, and it works just fine for a lot of things. Kai, you got louse. The Peneste angustipennis cognata, I gave you a louse. I do have Cambodia going very well too, but I gave you louse because I have had those for longer. Tennyson says, mine will occasionally nibble on dog food, but fruits and even pashy bug burger is eaten immediately. Even fish food pellets disappear faster than dog food, but the dog food is still just slower than other stuff I provide. So, I mean, also, if your colony of stuff likes one thing or a cast of things, then if you want success, just keep giving them what they really like to eat is another way. Easy Grows It. Hello, Easy Grows It. You look like a new face. Kyle, talk about skunk roaches and the locations you have. Why do I feel like maybe somebody, Kai may have put this person up to asking me about my Eurycotus localities. So I really like Eurycotus floridana because it's our native polyzosterine roach. And I think that's so cool that we have this link to Australasian fauna through this roach that is endemic to southeastern United States. And so in hopes that perhaps there's some unique traits hiding in these that could be isolated in captivity, but also just because, it tells a genetic story. I have multiple localities of Eurycotus floridana. The most distinct ones so far are Big Torch Key, which have the borders on the pronoda and on the thorax. And I think those are really cool. And I'm currently breeding for a line that has very well-defined borders because the first 3 individuals I received, one of them had very faint borders, and that still pops up in the line, but I'm not going to select against it because it's a natural phenotype. Hidden Lake is from southern Florida. And surprisingly, they resemble the Big Torch Keys more than they do the other mainland varieties of floridana, which suggests that perhaps they are more closely related. And that would tell a bigger story about perhaps sea level changes and whatnot in southern Florida that would have perhaps linked these populations at some point and then isolated them. And for whatever reason, this trait is advantageous in southern Florida to have these markings sometimes. Gulf Shores is a pretty northern locality of floridana, being in Alabama, maybe one of the northernmost limits perhaps for their range in the United States. Key Largo is pretty self-explanatory, but actually I think the Key Largo floridana tell an interesting tale in that they don't seem to be as vividly marked as the lower keys or even the mainland Eurycotus floridana, and I think that, as has happened with quite a few other keys species that have closely related taxa on the mainland, I think that these intermediate characteristics on the Key Largo Eurycotus floridana are evidence that there has been intrusion of mainland genes from further north onto Key Largo, probably brought in on landscaping materials or what have you, that would have caused an influx of mainland genes into them. And League City, Texas is another strain that's showing some promise to be quite unique. They seem smaller than all the other Eurycotus, and it's Eurycotus floridana from Texas. League City, I think, is pretty disjunct from a lot of other populations. Maybe they got there via timber or something in recent human times, or maybe it's just it's part of this is just part of the historic range that they've always been in. But so far, it might be one of the westernmost points for finding floridana. And if they are in fact smaller, and that's a consistent trait, that is a good suggestion that they are genetically very unique from the other floridanas, aside from being located so far away. And finally, Silver Springs is sort of your basic mainland, what a lot of people have been culturing for a long time, Eurycotus floridana with the solid bodies, which again is mostly a northern and apparently western phenotype, because the League City ones have those too. So anyways, easy grows, and I hope that was a good discussion on those. I think I'm pretty set for floridana localities. Maybe if I end up going back to not Miami, to Tampa, I might grab some from there, because that's a Western Florida locality, but I'm not 100% sure. 6 bins, not like they take up a lot of space, but 6 bins is quite a bit to devote to floridana, but who knows? I would like to get some from Louisiana and from Georgia, if possible, even. Madison says, I feel like I always learn something new listening to Kyle. So much knowledge. Well, thank you, Madison. I'm glad. Tennyson says, just realized how weird it is that I like learning about this stuff. I'm pretty sure I have read every single invert description. You guys really know how to flatter me. I appreciate that everybody here, or a fair number of people here, are keeping up on the site updates and stuff like that. I wish I could. I really wish that I could work on the site more, but I have to also run a business simultaneously, and I have to pack bugs and whatnot and keep on top of that too, which has been going great lately. But I think within the next 5 years, as Roach Crossing expands and moves into a permanent facility, I think that hiring some full-time staff who I trust to take care of bugs will open up opportunities for me to put more information on the website and make things more accessible to a lot of folks. So, but it will take time. It will definitely take time. Easy grows it. You're welcome for the explanation. I'm is quite happy to hear that you are not being facetious, because Kai often makes fun of me for my desire to have multiple localities of Eurycotus floridana. But again, it's really cool because they're an endemic U.S. polyzostrine. They're big, they're unique, they have all kinds of adaptations to deal with the southeastern United States, and I have my fingers crossed for maybe one more cool locality or something will pop up. Maybe something with different markings, or with again a different size or something like that. Something to make them very distinct from the other ones, and sort of fill out that sort of picture of this species adaptation to the southeastern U.S. Russ says from Aquariumex Pets. An update on my bioactive Grylotes sigillatus bins. Crickets are breeding plentifully. You said that bioactive cricket bins smell much better than other culture methods. I can confirm it. They really do. Just getting the springtails in there really cuts down on any odor. And very mild garden soil type smell. Yeah, I mean, that's probably the bacteria living in the soil, and there's some evidence that that they improve mood depending on what type of bacteria they are, which is certain types of soil bacteria give the post-rain smell that people tend to really like, and I think it's associated with elevated mood or decreased stress hormones or something like that too. It's very interesting. Missed it, and it smells like rail. Oh, there we go. I didn't even read that, Tennyson. So now you see you've got some indoor ecology going on. Buffet is daily. I'm running out of stuff fast. I don't feel like paying for different feeds. Yeah, that's fine. What's the deal with Eurycotus opaca? I don't see them available these days. I think I've switched a lot of my roaches to high ventilation, and it works great for a lot of roaches, but I think some species just want that extra humidity, and stuff in the basement, in the bug room, does not get tended to or babied as good as stuff up here does. So I did very well with Uricoris opaca for a long time, and then what was the first decline that started to happen? And then the first couple of moves happened, and that kind of took chunks out of the colonies, and then other life stuff happened, and that kind of finished some of them off. But Opaca Soroa I had going in fair shape until maybe last year-ish. The last time that I had a good number of Opaca was when I was keeping them with my Elliptorhina chopardi in a very large Rubbermaid bin with a heat lamp on one side. And when I think back to all the times, what? I think he heard a dog outside. Well, I think back to all the times that the Opaca did very well. It was always very high humidity or very limited ventilation, or even if there was good ventilation, it was still a covered top enclosure. So if I work with them again, because I'm tired of messing around. What? You want to come here? No? Okay. Because I'm tired of messing around, I still have the original containers that the cultures did well in. So I will literally just recreate the first enclosure that I kept them in and get results unless something's seriously wrong. So, but again, a lot of people. What's up? I think Victor wants me to end the stream, maybe, but I'll get through this, and then oh boy, we're at 2 hours already. This is a good stream. Good stream. Let me go through this. So I think that's why is they're just a kind of a finicky species with a very very high need for something specific, and a lot of us were not able to give them that at different points in time. I told you I didn't know that you had some Enrambifolia from a different strain. I have them in one of these enclosures. They seem to be doing pretty well. I think Rambifolia might have needs similar to Opaca, and so I have to fight my obsessive compulsive desire to keep all my roaches in high ventilated tanks and come up with a new lid for Opaca and for Rambifolia that will accommodate both the high humidity summer keeping style and the low humidity winter conditions in the bug room, because I'm not going to run a dehumidifier. I'm not going to run a humidifier in the winter time because it'll mess with all the upstairs of the house stuff, the window gaskets and all that stuff. But so anyways, I can re-bandage fine, but I can't. Oh, Dilo. So banded crickets, in my experience, buying them and trying to start them from pet store stock multiple times, they're just so stressed and or diseased that you just can't get them to go. In fact, I got some Akita domestica from a pet store, and I painstakingly picked out the eggs that they laid into a separate incubation enclosure, and then started raising up the nymphs, and they just started dropping one by one. There's some pathogen or something wrong with them that gets transferred even to the eggs, apparently, at least with Akita domestica, maybe with these sigillatus. And so I just had to start with wild stock. I started with 2 female wild Grylotes sigillatus and made the Florida Man strain, which is available on the website. I have no problems. But starting and when I've had pet store crickets get loose in my yard and breed over a summer, they do fine. So they're obviously able to just very meticulously find the right food and the right spots to sort of get past whatever pathogens they're harboring outside, but they can't do that in an enclosure indoors. So it was just easier to start with wild stock from Florida. So, but yeah, they're so ridiculously easy. But again, there's something wrong for small scale growing with the pet store crickets, and so that's why I now I have to find, when I go to Arizona, hope that I find wild Akita domestica, because I don't want to work with these diseased pet store crickets again. So banded cricket babies love to kill themselves on whatever moisture I give them, blah blah blah. You don't give them moisture then. Just give them a light misting every couple of days, especially at this time of year. You can maybe even do once a week. During the winter, if it's really dry, every couple of days or just once a day. And it doesn't have to be much. If you have a little hand sprayer, just one quick spray when you walk by there a day, and they can go and drink if they need to. Rocky pine scrub with high heat and seasonal rains or multiple scaper morphs. Dry scrub with small moist microclimates they can retreat to when it gets dry. Big body ATL. Can you show some hissing roach species? Yes, I think I'm going to show some hissing roach species. I will do that at the dead end of this stream, which is going on a long time, but is wonderful. I'm going to read through the remaining comments, answer some things, fetch a hisser, and then wrap things up because I do have to clean rodents tonight. So I will do that in one moment. Big body ATL. Do you plan to get more and breed them? Tennyson, do you mean the Grylotes? Because I have that colony going. Yeah, didn't you say you weren't sure if you'd have a lot to talk about at one point? Yeah, it really did change. This is a good pressure release valve today. See, for Rumble, it does seem like high ventilation, low humidity is the way to go. That's how the keeper I got mine from cultures, and they're thriving from, which is interesting though. Like, do they have a measure on the relative humidity in their bug room? Because it might be a lot higher. The air humidity may be very high, even if the enclosure humidity is low. Now, when somebody who's currently living in Hawaii has found rhombofolia, it was after rains. And they seem established in places that are quite humid, like southeastern Texas and Hawaii. Those are places that may have not sort of damp, stagnant humidity, but they have very high air humidity at different, well, Hawaii all the time, I guess. Cricket paralysis virus. Yeah, Alan, but the thing was, I thought I had read that cricket paralysis virus actually was found not to be responsible for the Akita problems, and it was a combination of some other things, supposedly. Some discussion at Entomobria unostrigata. This Saturday, I am taking some time out for myself, and by that, by taking time for myself, I mean I'm going out and doing more bug stuff. But my cousin needs some work done at her enchanted forest log cabin getaway, and so I'm going to go out there and do some of that work, and then go look for some stuff in her woods. She has a couple acres of property, and I wanted to get some mud minnows to introduce to my pond and some other stuff, and get some plants. There are some interesting plants there, and there's so much forest to explore. There will be some springtails, so I hope to add a couple more springtail species to the chamber, which I still have to move up there. It's a long story. Maybe I'll tell it next week. Yeah, ambient air humidity is rather high. So yeah, I think maybe in the winter time I might switch to a solid lid instead of the screen lid for my rhombofolia and whatnot, because they did fine all summer. Because right now in the base, what's the air humidity here? It is 58% relative humidity in this room right now, which is up from 38% just like a week or something ago. So yeah, all right. So it seems like I've answered quite a few questions and whatnot. So I'm going to move on to showing off a very special hisser. Very special too, hisser? So these are, maybe I showed off the male before, I can't remember. These are pure locality Gromphidorrhina Portentosa from Mosola in Madagascar. These are not wild-caught individuals, but they are locality strain of Portentosa, unlike anything we have ever seen before in the US, which I received from a lovely somebody who is currently in the chat at the moment. So I was for a couple of weeks, I was like, this female kind of looks mature, like she's big, and I like kept checking the underside of her abdomen, and the segment just didn't look right for an adult female. I was like, maybe, maybe not. Then this male matured, and I was like, oh yeah, yeah, that's a good male. And this female, she's gonna be that's an adult female. And then maybe like 3 or 4 days ago, the female molted again, and it's this huge monster thing now. It was not mature. um, this is, as TJ is saying, because he's the originator of the strain. It's a very pretty strain. My crappy lighting does not do them justice. There is sort of a very red orange coloration to them. I would describe the Clevelands as more orange brownish. Not saying that that's a bad thing, because I do find the Cleveland Portentosa very attractive. But also interesting is there's some stripes here on the first couple abdominal segments on both the males and the female, the male and the female, are multiples to look at. And the epaulets are pretty pronounced. The little epaulet markings on the thorax are pretty pronounced. So it's a very beautiful, beautiful strain of Portentosa, and will hopefully gain popularity, and hopefully the evil of the isopod hobby will not creep into the roaches and label all Portentosa as exactly the same, and that would ruin the beauty of this very unique strain of Portentosa. And having seen these in person, the mixture of colors and textures and whatnot really makes me rethink some perspectives on Gromphidorhini taxonomic relationships, and really thinking that maybe even considering the past experience I have with Ailuropoda hybridizing with Gromphidorhina princessia, that maybe they should all be in the same genus, and there should be some distinctions at subspecies or variety or something like that, or regional variation or something like that. There's something that Oren had talked about for a while. This doesn't mean that I promote the mixing of different strains as they're currently listed as different species and whatnot, but I do. The bottom line is we have all agreed upon and have known for quite a while now is that hissing cockroach taxonomy needs to be looked at very, very badly. And apparently it's being looked at currently, but it's not being published, or it's going to be, or it's not going to. I don't know. I want a paper that delineates everything using pure stock. So, Tennyson, I've only had them long enough to raise them up from about half grown to, or a little under half grown to adulthood. So I can't speak for how easy they'll be relative to the Cleveland Portentosa, but likely they're probably the same. Ranamophana Madagascar strain. Yeah, TJ, I wish you the best of luck with doing that, because that would just be a perfect double whammy to get all these. It's been so long since we've gotten new hisser strains or stock rating. The last one was the Mahoganies from Dan at Limberlost Exotics, and which are great. I love the Mahoganies, and of course, there's all the strains that I'm working on refining. But to get something wild, is phenomenal, because the last sort of truly wild stock that we received was Elliptorhina Davidi, and that was. Again, I just have to say this every time because of all the inbreeding stuff and all the things that people say. But all of our Elliptorhina Davidi come from a single female collected under a palmetto frond, and I was told that by the collector. So yeah, so this is great, and some of the other things that have popped up and that I'm working on. Some of you may remember the. I was this maybe a couple months ago or weeks ago. I was talking about how I got upset because I had picked out this pair of hissers for this breeding project that had what I thought was the perfect color, flagship color to create the strain using, and I put them into the enclosure. And then when I went to go check on them a week or a couple days later, I realized that I had not removed every single pre-existing adult male from that enclosure, and I was upset about that. But the female from that pair has produced offspring, so we're gonna see what the F1 of that crossing looks like, and if it looks like mom and dad and not like mom and dad number two. I'm going to be quite upset, but we'll see. It's gonna be it's gonna be a ways off, but there are babies produced. There should be baby Lilliputians on the way too, as well as some other projects. But we'll see. I'm really excited to get Lilliputians out there, just because, quite frankly, just for the meme, because of having a dwarf common. Do something about that common name. Dwarf common hissing cockroach be the same size as or smaller than a dwarf hissing cockroach is just quite amusing to me. So the Wild Martin, nice of you to join us, and unfortunately you're at like the tail end of the stream, and this is like one of the longest streams that I've done also. So it is great to see you here, but a lot of the content you may have to go back and revisit. So are hissers able to be bred for size? Tennyson, yes, I am currently working on that, both breeding up in size and breeding down in size with pure Portentosa stock. So, all right, so for big body ATL, I do have wide horn hissers as well. Available and just in general. So, but yes, hissers are wonderful and marvelous. It would be great to see different species and different color varieties from all across the island, because they are certainly a beautiful roach and what got me into roaches in the first place. So, with that being said, I'm gonna go ahead and put these beauties back and wrap up this lovely long live stream. It was great getting to rant with all you guys. Great seeing some new faces and seeing a lot of old faces return as well. I will see you all next week in the lovely month of June, hopefully with some interesting finds from my cousin's place, and maybe with some good news from the colonies and whatnot as well. So, so yeah. Take care, everyone, and may your cultures be ever bountiful. And try and wave goodbye, but I don't want to irritate her. So, good night.